Good evening, all. Hello.
Good evening, sir.
Is it audible to you? Am I audible?
Yes, sir. Yes, sir.
Screen visible to you?
Yes, sir.
Hello? Right. Today's topic is carbohydrates. Thought in it. So we know the carbohydrate is a large biological molecule, and it is made up of carbon, nitrogen, and oxygen. What is the formula of general carbohydrates of, for example, a glucose or glucose structure? If you take actually glucose, best example, glucose is just a carbohydrate. Glucose formula C6H12O6, right? So majority they maintain in carbohydrate the carbon and hydrogen oxygen ratio 1:1 ratio, right? They're extremely widespread in plant, compressing up to 80% of dry weight of plant body. Is it clear or any disturbance?
No, sir.
Any disturbance?
No, sir.
Right. So they are most abundant organic molecule in nature and also referred as saccharides, right? So general name, these are called these carbohydrate in scientific name, scientifically called as saccharides. The carbohydrates which are soluble in water and sweet in taste. But some carbohydrates non-sweet in taste. So actually there are two types. The carbohydrates, two types, sugar and non-sugar. So not given here. I will explain in classification. They are organic compounds and organized in the form of aldehydes, mainly aldehyde and ketone group. Aldehyde group and ketone group with multiple hydroxyl group coming off the carbon chain. So take classification. These are mainly classified into three types. One is monosaccharide, second oligosaccharide, and third one polysaccharide. Before that, actually, as I said earlier, these carbohydrates two types: sugars, non-sugar. Sugar which are sweet in taste and soluble in water. Best example, glucose and fructose, right? Hello? Glucose
Yes,
and
sir.
fructose.
Yes, sir. Yes, sir.
Non-sugar, these are tasteless. That means they are not in sweet taste. They are insoluble in water. Starch, cellulose. Starch and cellulose. Now in classification, first one, monosaccharide. What is mono? Mono means single, right? Mono means single.
Yes.
So these are the simplest carbohydrates. Actually, carbohydrates, when it is hydrolyzed, these are split into, degrade into simple sugars. But monosaccharides, they cannot degrade, they cannot separate. When we dissolved in water, cannot be hydrolyzed into simpler carbohydrates because they are already in simple structure. Example, glucose, mannose, galactose. They are very simple structure. These are often called as a simple sugar, right? Example, glucose, mannose, galactose. What about oligosaccharides? When we hydrolyzed this type of sugars, this type of carbohydrates, they are degrades into different sugars or different type of monosaccharides. It gives monosaccharide molecules. These are compound sugar that yield 2 to 10 molecules of the same or different monosaccharides. If it is same type, that is called homo. If it is different, that is called hetero. Same homopolysaccharide and different heteropolysaccharide. The same homo oligosaccharide, different hetero oligosaccharide. So that is the based on the chemical structure of sugars, right? Maybe this oligosaccharide may be 2 to 10. It gives, if it is only one type of sugar, that is monosaccharide. More than one, maybe two, di, three, tri, like that, it up to 10, maybe up to 10. So here given some examples for disaccharides. Here the disaccharide example, disaccharides, sucrose, lactose, and disaccharides, raffinose, and tetrasaccharides, stachyose. So these are the different type of, and in our book given different type of, again, then the disaccharide, sucrose, maltose, lactose, cellulose, tahallose, and trisaccharide, raminose, getinose, and raffinose, tetrasaccharide, tachyose, and pentasaccharides, verbose. So these are the different type of sugars. If it is exceeded, when we carbohydrate hydrolyzed in water, if it is exceeded more than 10, that is considered as a polysaccharide. Poly means number of. So only one monosaccharide, more than 1 up to 10 oligosaccharide, more than 10 molecules of sugar. That means it gives different or same 10 above monosaccharides. It gives 10 above monosaccharides. These are called polysaccharides. The best example, starch and cellulose and glycogen. Right? Actually, which type of food material synthesized in plant body? Hello? What is the reserve food material in plant?
Starch, sir.
Starch.
Yes.
But actually, before formation starch to photosynthesis, what are formed in plant body?
Glucose.
Glucose or
Yes.
fructose.
Yes, sir.
Maybe simpler form or disaccharide form. But later, these are all together accumulated into polysaccharide and stored in the form of starch. Right? And what about animal cell? In animal cell, the same, the glucose. Here also we take for energy glucose, but excess glucose converted into liver as a glycogen. Right? So here the polysaccharide two types. Homopolysaccharide means starch. Starch means here if we hydrolyze to starch, it is formed a number of glucose molecule. Number of glucose. These are all same, but not other molecule, which is made up of completely one similar type of molecule, that is glucose. That's why this is homopolysaccharide. And glycogen, inulin, cellulose, chitin, pectin, they are all same type of, made up of same type of monosaccharide. Now heteropolysaccharide, hetero means which are made up of different type of sugars. Example, gums and hemicellulose. Gums and hemicellulose. So these are the different type of sugars. The types of carbohydrate, classification of carbohydrate also important, but in our book not given sugar, non-sugar, just add it. Sugar and non-sugar, sugar, glucose, fructose, and non-sugar, starch, cellulose, enough. After that, you go through this monosaccharide, oligosaccharide, and polysaccharide. Right? Apart from this classification, just explain about the structures. Structures of monosaccharide, that means structure of glucose, mannose, galactose, any one and enough. Give the example glucose, right? Draw the glucose structure. Next, oligosaccharide. In oligosaccharide, so disaccharide, one example enough. Tetrasaccharide, tetrasaccharide, one example enough. Diagram. And right, tetrasaccharide, one example enough. Penta, no need. Verbinose, no need. 2, 3, enough. And here polysaccharide also, starch, one example enough. Right, let's see the chemical structure of the oligosaccharide, because already glucose structure we know. Do you know or not? Hello? Do you know glucose chemical structure?
Yes,
Hello?
sir.
Right.
C6H12O6.
Right. Now just go through this oligosaccharide. These are the oligosaccharide. Oligosaccharide, first let me complete disaccharide, later trisaccharides. First one, disaccharide maltose. Disaccharides, these two, one example, one enough. So the first one, maltose. Maltose made up of, these are formed by, first of all, the oligosaccharide. These are formed by union of two monosaccharide. Here when oligosaccharide, I am explaining about only disaccharides. Disaccharide formed by union of two monosaccharides. This is one, this is two, first two. So two monosaccharide. They are held together by glycosidic bond. This bond, this is one. This bond called as glycosidic bond. Glycosidic bond between the first carbon of monosaccharide, first carbon. This is already given the number. This is the first one. First carbon of one sugar with fourth carbon of another monosaccharide. This is here fourth. Fourth carbon of another. So this is glycosidic bond. The disaccharides are two types. Maltose Lactose. In our book, give two example: maltose and lactose. You can draw any one, not both. Any one you can draw. So first of all, let me see. First, maltose, malt sugar. The maltose made up of two alpha D glucose in it. This is one D glucose in it. I will explain later what is D glucose and L glucose. D glucose. D glucose. D glucose to D glucose units. Right? So here the linkage bond formed between the glycosidic bond 1 to 4, first carbon and fourth carbon. Right? This maltose is produced when starch is hydrolyzed by amylase or by acid hydrolysis. So this maltose produced when starch hydrolyzed. If it starch hydrolyzed, this maltose produced. Right? So it is made up of two glucose molecules. This one glucose and this is second glucose. Right? Next one, lactose. The lactose, which is called as milk sugar. Lactose called as milk sugar. Milk sugar, it is found in the milk of mammals. It is formed by the union of a molecule, same D-glucose, and one molecule of D-glucose by galactose. Galactose, sugar, this is monosaccharide by D-glucose. Glucose and galactose. The same here also glycosidic bond. Here also same glycosidic bond. This is also first carbon. This is fourth carbon. Right? Is it clear? Hello?
Hello, sir.
Right. Next.
Hello, sir. What's happening?
Just wait. Right. So this is the example of trisaccharides. Trisaccharides, raffinose. First example, this is the raffinose structure, which is made up of galactose, glucose, and fructose. The most common trisaccharide found in plants. It is found in plants. Raffinose. Raffinose, which is trisaccharide. That's why it is made up of three monosaccharide. One is galactose. Another one is glucose. Fructose. It is present in cotton seed. Cotton seed. This type of sugar mainly present in cotton seed and sugar beet. When it is hydrolyzed, the yield of raffinose, glucose, fructose, and galactose. Right? So this is, and next one, tetrasaccharide. Tetrasaccharide example, stachyose, which is made up of 4 monosaccharides. First one, which is galactose. galactose, galactose, second also galactose, first sugar galactose same, and this second also same galactose, and third one glucose, this one glucose structure, glucose, and fructose, last one fructose. Fructose called as fruit sugar. Fructose called as fruit sugar. Cane sugar. Hello.
Hello, sir.
What is cane sugar?
Sucrose.
Sucrose, cane sugar. Milk sugar, lactose. Fruit sugar, fructose. Right? Okay. So this is oligosaccharide. And if you want to draw the polysaccharide, in polysaccharide, one example enough, that is the starch. If we want to give the starch diagram, this glucose molecules, this glucose chain, the same, this glucose chain, the number of glucose chain continuously, number of uncountable glucose polymer made up of, that is starch. That is called starch. So that is why polysaccharide. It just to turn on page number 41. 41. 41. The first here homosaccharide, starch, glucose, cellulose, and chitin also. And heterosaccharide here, gums and hemicellulose mucilase. First, let me complete homopolysaccharide. Starch, already told you, it is made up of sugar. Right, D-glucose sugar with the bond, glycosidic bond with 1 carbon to 4th carbon. So 1,4 glycosidic linkage. 1,4 glycosidic linkage. And what about glycogen? It is storage form of carbohydrate found in liver and muscles. In animals, excess of glucose stored in muscles and liver, and there it is converted into glycogen. Right?
Okay, sir.
Right. And here the bond formation, not 1 to 4, but here the glycosidic bond formation in glycogen. In glycogen, the glycosidic bond, first carbon of one sugar, 6th carbon of another sugar, 1 to 6 linkage, glycosidic linkage. And next one, cellulose. Cellulose, where is cellulose? Where is cellulose? Cellulose chemical structures of. Where do you find out the cellulose?
Cell wall. Cell wall, sir.
Cell wall. So the cell wall mainly present in animal cell or plant cell?
Plant cell.
That's why in plants most abundant biopolymer is cellulose, and which is made up of the same 1,4 glycosidic bonds. 1,4 glycosidic bond with the same molecules. That means same monosaccharides. And chitin, where the chitin? Chitin.
In animal cell structures like
Cell structure.
sorry.
Which cell structure? Fungi cell wall. fungi cell wall made up of chitin. The chitin monosaccharide of D-glucosamine, which is linked with same 1 to 4 glycosidic bond. Right? Next, dextran, fructosanes, they are all example, but chitin enough. So in homosaccharides, homopolysaccharide, starch, glycogen, cellulose, and chitin enough. No need to write about dextran and fructosanes. And heteropolysaccharide, page number 4.2. Heteropolysaccharide, hetero means same, here also more than 10 sugars, but they are different type of monosaccharides, not the same. In a chain, maybe glucose present, fructose present, galactose present, so different type. Is it this raffinose, homo or hetero? Raffinose, homo or hetero?
Homo, sir.
Raffinose.
Homo.
How it is homo? Galactose, glucose, fructose, which is made up of 3 different monosaccharides. That's why it is hetero. If it is homo means which is made up of all type, one type of all made up of, which may be made up of glucose, or it may be made up of galactose, or maybe made up of fructose. That is homo.
Okay, sir.
In heteropolysaccharide, best example, gum. The branched structure containing D-galactose, L-arabinose, L-drominose, gluconoic acid. So different type. Here hemicellulose also D-glucose, D-xylose, arabinose, gluconoic acid. And another heteropolysaccharide, mucilage. Your mucilage also made up of different type of sugar monosaccharides, D-mannose, D-galactose, and sulfuric acid also. And here with sugars, other molecules also present. And clearly given the table about polysaccharide, homopolysaccharide, storage and structural. The storage, best example, inulin, starch, animal glycogen, structural plant cellulose, animal chitin. Animal here the best example given is the fungi. Chitin main structural unit of fungi. Heteropolysaccharide, gum, mucilage, hemicellulose. Right? What are the functions of the carbohydrate? What is the best first function? Carbohydrate function.
Give energy, sir.
Energy, that is the first fundamental one. Gives energy. A nutrient to the most living organism. In commercial, it is used as a food and sweetener. As a carbohydrate, we can use it as a sweetener. And already given the repeated, so sucrose provides substrate for energy. Not only sucrose, for glucose, fructose, sucrose, anyone. Give the substrate to the energy production. Already we learned in respiration. In respiration, glucose glycolysis, Krebs cycle, finally production of ATP energy. So in any type of cycle, the first fundamental is carbohydrate. Substrate is carbohydrate, whether it is fructose or sucrose or glucose. Right? And starch is the principal reserve food material, like green plants. Here given the example, cereals, potato, legume, other vegetable. And these carbohydrates act as the structural units in plants and fungal member and bacteria. So if we take the plant cell wall, cellulose, which is made up of carbohydrate, right? If we take fungal cell wall made up of chitin, which is also carbohydrate, right? And if we take the bacteria cell wall made up of peptidoglycan, which is also carbohydrate. So these are the importance. Without carbohydrate, the plant cell can protect with help of cellulose. Correct? Protect. Hello?
Yes, sir.
Can the plant cell be protected without carbohydrate?
No, sir.
No. That's what these are all important. And this cellulose, another important in industrial purpose, making paper, cloth, paper. How to make paper and how to make cloth? Give one example for cloth. Cotton made up
Cotton.
of, right, cotton.
Yes, sir.
And any other importance? And this carbohydrate utilized as a food material. That is also we can include vegetables, rice, flour, wheat, jowar. They are all the main nutrition. Rice and jowar, in the anything, the carbohydrate, the main nutrition. That's why we have been using this as a food source. That is also one function. And any other bacterial dextrosan, pectin, which are present. Okay. Right. So this is carbohydrate functions. Okay. And next one. Structural aspects. Structural aspects. Asymmetric carbon atom and isomerism. Right. Asymmetric carbon atom means the carbon asymmetric carbon atom. Page number 3333. For the given asymmetric carbon atom. Asymmetric carbon atom, a carbon atom which is made up of four different atoms or groups. Four different atoms or group that is called as a asymmetric carbon atom or chiral. Example. See, different atoms. Fluoride, hydroxy, 1L, Br bromide, hydrogen. So this is called asymmetric. This is called asymmetric. Right? And next one, isomerism. What is isomerism? Simple definition. What is isomerism? Have you heard this word? Have you heard this word?
Same formula, different properties, sir.
That's it. That's
Same.
it. That's it. So two or more compounds with the same molecular formula. Molecular formula same, but the arrangement of group difference and physical chemical properties of different.
Yes,
So
sir.
that is called isomerism. It is called isomerism. So formula same, but the functional group formation different. Functional group formation different and physical chemical properties also different. That is called as a isomerism. In isomerism, the mainly different type of isomerism. These are first of all these chemical compounds. This compound, different compounds and same compound. The same molecular compound, same molecular compound. Are you follow my cursor?
Yes, sir.
Yeah. Same molecular formula, that is called isomers. Same molecular formula, isomer. And second one, different that is different compounds. And the same connectivity atoms. The based on connectivity atoms, two types. That means these isomers, two types. One is stereoisomer. Second one is constitutional isomer. Or structural isomer. Structural isomer means here the different is the first in, for example, let me draw here. First complete it. First one I will explain later. Next in stereoisomer again on configurational isomer and conformers. These are interconversion breakage bonds. Based on breakage bonds, again two types. And based on mirror images. A mirror images present enantiomers. Mirror images present enantiomers. Absent diastereomers. So these are the isomer types. Okay. First of all, let me complete structural isomer. So isomer definition clear, same formula. But in based on connectivity, the same connectivity atom, that is stereo. Different, that is structural. First of all, let me complete about structural isomer. Structural isomer means here the plant given. Where it is glucose, fructose, not given example. Just wait. Structural isomer, example glucose, fructose. What is formula of glucose? Hello?
C6H12.
What is formula of fructose?
Same.
Formula same, right? What?
Yes, sir.
As I said, the connectivity of atom. Same connectivity. If it is same connectivity, stereoisomer. Different connectivity, structural. Structural example, glucose, fructose. Let me draw here the glucose formula C6H12. So here the 1, 2, 3, 4, 5, 6 carbon. Glucose, keto group. That's OH, hydroxy group. Here hydrogen. Left side OH group. Here hydrogen. And next. Right side OH group. Left side hydrogen. OH group. Here hydrogen. Last CH2 OH. So this is glucose formula. If we take the fructose formula, the way the right. So keto group first carbon here. The keto group first carbon. If we take the fructose formula, first CH2OH, first carbon CH2OH. Second carbon. This is second carbon, right? The keto group present at second carbon. It's double bond O, second carbon. Rest of the all same. Rest of this structure, the remaining chemical structure same. So what is the different here? The connectivity. Is it same connectivity or different connectivity? The keto group, C double bond group, first carbon in glucose. Whereas in fructose, second carbon. That is the different connectivity. If it is same connectivity, that is called stereoisomer. Okay? Hello? Hello?
Yes, sir.
Is first one clear? Constitutional structure isomer clear?
Yes, sir.
Right. And next one, stereoisomer. Stereoisomer interconversion require breakage bonds. First of all, configurational isomer. Just wait. Configurational isomers. Okay, conformers not given. Okay. So in our book given D-glucose, L-glucose. So stereoisomer here. The structural clear. Stereoisomer D-glucose. Two, three, 4, 5, 6. Here the fifth carbon, H, OH. HOH. That means here the OH group right side. That is in D-glucose. D-glucose. In L-glucose left side. L-glucose left the same structure. No change. If the connectivity same. If you take structural connectivity different, one is first carbon, one is second carbon. But if you take the D-glucose and L-glucose, this is D-glucose. In the same structure, if it is L-glucose, only change is this OH group, not right side. The OH group should be in left side, this side. Is there any changes in connectivity? Hello? Hello?
Yes, sir.
OH group connectivity, any change? That is carbon
Yes,
number, any change?
Side is different.
One is right side, one other one is left side.
Left side. Yes, sir.
That is the D-glucose and L-glucose. One example enough. In our book, 2 example given, but 1 example enough. In our book, not given the structural isomer. Just draw the structural isomer, glucose, fructose. Glucose and fructose. This is the glucose structure. The first completed structure, glucose, and second incomplete structure, fructose. What is incompleted? Only I will have explained only difference between connectivity. First carbon ketone, second carbon ketone. Third carbon same as it is. OH, H. And fourth carbon same. Fifth carbon same. Sixth carbon same. No change. So the change is only first carbon, second carbon. But in isomer, Stereoisomer, one example enough, no need to confirm configurational and conformations. And one example enough stereo, in stereo D-glucose, L-glucose. The same structure, same connectivity, hydroxy group, one is right side, one is left side. That's it. Is it clear? Hello?
Yes, sir.
Stereoisomer also completed. Next one, actually in our book given, that is optical, where it is? Optical isomerism. In optical isomerism, again, these are two different enantiomers and diastereomers. The same enantiomers and diastereomers, this is the, if it is same mirror image, complete structure, not one carbon, two carbon. The entire structure mirror image, that is called enantiomers. It's not follow the mirror image, that is called diastomers. Best example here, not given book arabinose, ribose, HO. Just wait. Structures of D-glucose. In optical isomerism, another important is, in optical isomerism, for example, this is the water, sugar, dissolved sugar. When we pass the light through these sugars, it turns into, one is clockwise direction or anti-clockwise direction. It shows the clockwise and anti-clockwise direction, that type of isomerism called as optical isomerism. So with the chemical, the dissolved chemical inside the water, when we pass the light, when we using the light, automatically these sugars can take into clockwise direction and anti-clockwise direction. This type of property is called as optical isomerism. Right, in this optical isomerism, enantiomers. The best example, enantiomers, actually, can I just wait? Is there any diagram? Okay. For example, this is glucose structure, right? Hello?
Yes, sir.
Just this structure show in mirror.
Glucose.
Let me just wait. The same diagram, if you see in mirror, this O group, right or left?
Yeah, right. When we see in the mirror, it looks.
Right? For example, here OH group. Here H group. So now this is a diagram A, this is diagram B. In diagram A, OH right side. When we see in mirror, it is left side. That is the mirror image. The type of, if this type of mirror image present, that is called enantiomers. Are you clear about this? Hello?
Yes, sir. Clear.
In our book, we are given the best example, page number 35. Where it is? D-glucose, D-glucose. Actually, diagram not given. So anyway, I will give you, just wait. Are you able to see? Hello?
Yes, sir.
So this is enantiomer exam. CS3, OH. Right? And another one example. This is better. Very simple structure. D lactic acid, L lactic acid. Draw this. Take photo. Okay? Next, diastereoisomers. The best example, let me don't do. Wait. Is this mirror image? Heroes, electros.
No, sir.
Heroes, D erythros. COH here. Take photo of this one also. COH carboxyl group, here carboxyl group, same. But this is same mirror. Carbon H hydroxy. But here hydroxy. If it is enantiomer, here this side should be OH group, but not follow the same. So different. That means here the H group, here H, here hydrogen. Hydroxy. This is not a mirror image. This is not a mirror image. Right? It's clear?
Yes, sir.
Now carbohydrate, any remaining portion here, isomerism completed. Here given number of chemical structure, no need to draw all this. Epimers.
Sir,
Page number 36, epimers. The same. Diastereomers called as epimers. Diastereomers, another name called as epimers. Don't get confused. This is same. epimers. చెప్పండి. Hello? Someone try to ask question. Hello? 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 Hello?
Yes, sir. Listening.
Any doubt?
No, sir.
Clear? Right.
Yes, sir.
And now next unit. Carbohydrate clear or any doubt in carbohydrate?
Okay, sir.
Right, functions they are computed. Now the fourth unit, lipids. What are the lipids? These are the heterogeneous group of compounds. Is it soluble or insoluble in water? Hello?
Insoluble, sir.
Mainly they are the hydrophobic in nature. What is hydrophobic and what is hydrophilic nature? Hydrophobic, hydrophilic. Hydrophobic, hydrophilic, water loving, hydrophobic, water hate. And then maybe these are insoluble in organic solvent like acetone, chloroform, pentone, hexane. Mean this lipids insolubility most part of the water. Right? Maybe these are different form of fats, lipids are present. After digestion, they are converted into, in human beings, these lipids convert into, hello?
Fat.
This lipid conversion, synthesis of lipids, how the formation of lipid in plant body or animal body?
Fats, sir. Plants, sir.
How they are formed in plant or animal? In animal, by taking food, by taking fat, they are directly formed. Animal, in plant body, the excess of carbohydrates converted into fats. Excess of carbohydrates can convert it into fats. In animals and plants also. Right? So
Yes.
in food material, the storage food material, apart from the starch, based on the plant species, mainly inside the seed, they are stored in the form of lipids. Right? So here the most important one is beta oxidation. Very, very important question in theory and comparative level. And here given the different type of fatty acids, classification of fatty acid, classification fatty acid, like fatty acid, glycerolipids, and saturated fatty acid, unsaturated fatty acid, monounsaturated, polyunsaturated. Mainly this unsaturated fatty acid we get from the plants. Most of the plant body, the fats in the form of unsaturated fatty acid, which is beneficial to us. Saturated or unsaturated, which is good cholesterol. Unsaturated, unsaturated, good cholesterol. That's why the frequently doctors suggested only focus on vegetables. Who infected with heart diseases, who suffering from heart diseases, they are suggested by doctor only for vegetables, because in vegetables, in plants, unsaturated, mainly polyunsaturated are present. They are helpful to heart. Okay, and this is the types of lipids: saturated, unsaturated. Here also giving example: palmitic acid, oleic acid, choline, inositol. So different type of fatty acids, right? And here the important one is lipid mobilization, conversion of lipid to carbohydrate. Conversion of lipid to carbohydrate. That is most important one, right? Conversion of carbohydrate. Are you able to see?
Yes, sir.
Just wait.
Yes, sir.
Is it visible to you?
Yes, sir.
What about the screen?
Template.
Zoom screen also appeared or not? Zoom screen.
No, sir.
ఓన్లీ డయాగ్రామ్ ఒకటే చూస్తున్నారు, మిగతాది కూడా కనిపిస్తుంది డిస్టర్బెన్స్ గా.
All slides.
All 11 slides are seen, sir.
All slides are visible, sir.
Clear now, sir.
కింద ఈ బార్ కనిపిస్తుందా? జూమ్ యాప్ తో ఉన్న అప్లికేషన్ కనిపిస్తుందా? ఇదేం కనపడుతుందిగా మీకు?
No, sir. Only picture on.
ఓన్లీ పిక్చర్ కనిపిస్తుందిగా, రైట్?
Yes, sir.
Mitochondria.
Right. Now topic is fat converted into carbohydrates. So these are the fats actually mainly inside the seed. The fats are stored. These fats in oleosome convert into triglycerols. These are the triglycerols converted into triglycerols, right? Fatty acids are metabolized into bioxidation. This is next step. So with help of lipase enzyme, this is lipase. Are you follow my cursor?
Yes, sir.
So with help of lipase enzyme, the fats in oleosome converted into triglycerol. Now these triglycerols again convert into fatty acids. Fatty acids. Just wait. Now the fatty acids enter into peroxisome. This organelle is called, sorry, glycoxisome. This organelle called as glycoxisome. This is oleosome. One is completed, and this is mitochondria. So
Yes, sir.
Mainly to convert of the lipids, carbohydrate, utilize 3 cell organelles. That is important. What are these 3 cell organelles? One is polyosome, second glycosome, third mitochondria. Let's see the steps in each and every cell organelle. In first in oleosome, already the storage lipids are present in oleosome. They are stored in the form of fat or lipids. These lipids with help of lipase enzyme converted into fatty acids. First triglycerides. These triglycerides next converted into fatty acid. After formation this fatty acid, the fatty acid transported into the glycosome, right? Where these fatty acids formed as a acetyl coenzyme A. And this acetyl coenzyme A enter into the beta oxidation step. I will explain later beta oxidation complete view. So through beta oxidation process, this is oxidation process again formed as a acetyl coenzyme A. Now acetyl coenzyme A, right? After formation acetyl coenzyme A, now it is enter into the glycosylate cycle. In glycosylate cycle, same like a Krebs. Do you know Krebs cycle? Hello?
Yes, sir.
The same which follow the Krebs cycle, but Krebs cycle takes place inside the which cell organelle?
Mitochondria.
Mitochondria. Right, mitochondria matrix. But here the inside the glycosome, glycosylate cycle takes place. Right?
Yes, sir.
Where it is?
Glycolysis also take place in mitochondria.
Right. Before that, these fatty acids are oxidized. After oxidized, formed as a acetyl coenzyme A, right? Next, after formation, acetyl coenzyme A. Which received by oxalo acetate OAA with help of citrate synthase, citric acid form. Citric acid next convert into isocitrate. This isocitrate next formed as a succinate and cleaved. That is cleaved into 2 molecules. One is succinate, another in glycosylate. This glycosylate add with coenzyme A formed as a malate or malic acid. And malic acid enter into the oxidation process, again form oxalo acetate. This cycle called as a glycosylate cycle. And this cycle called as beta oxidation cycle. So beta oxidation cycle, glycosylate cycle, the both are formed inside the glycosome. Right? And why it is called oxidation? Oxidation means here we can see here NAD. Utilize hydrogen. Take hydrogen, convert into NAD or NADH2. That means from the acetyl coenzyme A, hydrogen removed, and this hydrogen received by NAD. Convert NAD. Here the same. The another hydrogen from the acetyl coenzyme A, one hydrogen take by NAD, convert into FADH2. So that's why it is called as oxidation. And here also, in this also, one oxidation process takes place. In beta oxidation, 2 oxidation reaction. In glycosylate cycle, 1 oxidation process takes place. Right? So anyway, here formed oxalo acetate. Oxalo acetate, take acetyl coenzyme A, convert into citric acid. Now the citric acid with react with water and convert into isocitrate. So here water missing here. Next, this isocitrate cleaved in the form of isocitrate lyase with help of lyase enzyme It converted into two different molecules, glycylate and succinate. Glycylate undergoes two oxidation process and formed. It is involved in again formation of oxalo acetate. But the succinate, they enter into the mitochondria, where it is oxidized into humarate. Oxidized into humarate. And again oxidized into malate, malic acid. Already we know the Krebs cycle, succinic acid, humaric acid, malic acid. Now this malic acid, malate, now that transferred into cytoplasm or cytosol, where it is again oxidized and convert into oxalo acetate. Oxalo acetate utilize ATP energy and convert into phosphoenol pyruvate. Through reverse glycolysis, fructose 6-phosphate. Finally, sucrose or carbohydrate form. Now the seed germinated. Now seed germinated means after formation of sucrose, what happened? If you want the seed germinated, this sucrose enter into the respiration process. And respiration means glycolysis, Krebs, ETS, again ATP energy form. So this is conversion of lipid. Here the lipids, first what is the substrate? Lipid. Lipid present in oleosome, converted into fatty acid. Finally, this fatty acid through the various steps finally formed as a carbohydrate. It is fats or lipids converted into carbohydrates. Is it clear? Hello?
Yes, sir.
Right. Second, next very important one, beta oxidation process. Beta oxidation. Beta oxidation. Very, very important one. Beta oxidation process. Beta oxidation process, a process by which fatty acids are degraded by oxidation of beta carbons and removal of successive 2 carbon fragments from the carboxylic end of fatty acid. Simple. What happened in beta oxidation process? Beta oxidation process from the fatty acids, Fa means fatty acid. From the fatty acid, from each steps, 2 carbon fragments are removed. 2 carbon atoms are removed. So this process called as beta oxidation, and finally remain as a 2 carbon atoms. First, let me start with fatty acid. First, fatty acid. Fatty acid may be 2 carbon or maybe 4 carbon, 6 carbon, 8 carbon, 16 carbon. So different number of carbons. But here I take here the one example, palmitic acid. Palmitic acid, which is made up of 16 carbon. Palmitic acid, which is 16 carbon. Palmitic acid, 16 carbon. What is our concept? This fatty acid finally convert into 2 carbon only. Convert into 2 carbon. Finally remain as 2 carbon. Right? For this, it is involved, take the 4 steps. First step 1, fatty acid activation. Second, activation of fatty acyl coenzyme A. Third, addition of water to the resulting double bond. Fourth, oxidation of beta hydroxyl coenzyme A, thiolytic cleavage by Koch enzyme. So these are the steps. Let me complete first one. The first fatty acid activated. In glycosome, fatty acids are activated by fatty acid thiokinase ATP and coenzyme A, resulting fatty acyl coenzyme A form. In glycosome, with the help of ATP energy, coenzyme A, convert into FA, means fatty acyl coenzyme, converted into fatty acyl coenzyme plus AMP common. This is the first step. Next step, oxidation of fatty acyl coenzyme. In first step, fatty acyl coenzyme A formed. Now this fatty acyl coenzyme A undergoes to oxidation process. What happens in oxidation process? FAD convert into remove hydrogen from substrate. This hydrogen taken by FAD and as a result convert into, hello? FAD convert into?
FADH2, sir.
That's FADH2. FADH2. And the product is trans to enyl coenzyme A. And FADH2 form. Right? Next, addition of water to the resulting double bond. Now, after formation, trans to enyl coenzyme A, there is no just addition of water converted into L beta hydroxy Please note down these steps. Hydroxy acyl A C Y L acyl coenzyme converted to acyl coenzyme. Next step, oxidation of beta hydroxy acyl coenzyme. Notice oxidation of beta acyl coenzyme. What happened? Oxidation of beta hydroxy coenzyme? Same. Now here the NAD, there is no place. NAD. First FAD, here NAD. NAD convert into NADH2. finally formed into beta keto acyl coenzyme A. Beta keto acyl coenzyme A. Now, last step, thiolytic cleavage by Koch enzyme. Finally, beta keto acyl coenzyme A cleavaged into fatty acyl coenzyme A and acetyl coenzyme A. Cleavaged into fatty acyl coenzyme A, fatty, say like this, fatty acyl coenzyme A and acetyl coenzyme A. Acetyl coenzyme A formed. So fatty acyl coenzyme A enter into the beta oxidation cycle. This is beta oxidation cycle, right? Hello? This is beta oxidation cycle. We start from the fatty acid. Again fatty acyl coenzyme A. After that, again this fatty acyl coenzyme A formed. For this, every step, two carbons are removed. But here, acetyl coenzyme A, how many carbon? Hello?
2 carbons.
Here, how many carbons in acetyl coenzyme A? 2 carbon. You take here 16 carbon, right? Hello? After
Yes,
first cycle, how many carbons remain here?
16.
First actually is 16. If one different type of fatty acids are there, but we taken one of the fatty acid, that is palmitic acid. Palmitic acid made up of 16 carbon. Now 16 carbon through these different type of steps finally converted into again fatty acyl coenzyme A and acetyl coenzyme A. This acetyl coenzyme A 2 carbon, already 2 carbon removed. And another one is fatty acyl coenzyme A formed. Now this fatty acyl coenzyme A and again enter the oxidation cycle. Same process. And again formed as a 2. That means after formation beta keto acyl coenzyme A, it is cleaved into
fatty acid acyl coenzyme A and acetyl coenzyme A.
right, fatty acyl coenzyme A and acetyl coenzyme A. That means in this step also 2 carbon loss. Every time in acetyl coenzyme A, 2 carbon loss. So up to how many cycle it takes to complete it? First of all, here the 16 carbon, right? In first cycle, it becomes 14. Okay. Second cycle, it becomes 12, 10, 8, 6, 4, 2. So what is our definition? Only it remain 2 carbon, up to remain 2 carbon, right? 1, 2, 3, 4, 5, 6, 7 cycles taken. That's it. Are you clear about this? Hello?
Yes, sir.
Beta oxidation, very, very important. In theory and comparative level. In lipid, most important question, the types of lipids, that is the general one, saturated, fat, and unsaturated, that is general. That's why I did not explain very much. Just only focused on conversion of lipids to carbohydrate, that is also important. And beta oxidation cycle, that is also important. These two very, very important, okay? In theory and comparative level. Are you clear about beta oxidation cycle?
Yes, sir.
In our book, what they given here? Right, in our book also given clearly, very clear steps. Page number 54. Page number 54, clear cycles.
Okay, sir.
So last, we can observe acetyl coenzyme A. How many acetyl coenzyme A formed? First cycle, one acetyl coenzyme A, second, third, fourth, fifth, sixth. Like different type of acetyl coenzyme A formed. That means in every cycle, 2 carbons are lost in the form of acetyl coenzyme A. Right? Then after formation, this acetyl coenzyme A, enter into the, hello, this acetyl coenzyme A.
Beta oxidation, sir.
Right? No, beta oxidation. This acetyl coenzyme A, this formation is this acetyl coenzyme A enter into glycosylate cycle, which is useful for conversion of lipids to sucrose. Lipids to sucrose, right? So this is beta oxidation cycle. Already told you when I explained the conversion of lipid to carbohydrate clearly. One is beta oxidation cycle. This is very important. Acetyl coenzyme, different type of oxidation there, but here not given any intermediate product. But in beta oxidation, I have explained clearly what are the intermediate product formed. Right, acyl coenzyme A formed and acetyl coenzyme A formed. This acetyl coenzyme A enter into glycosylate cycle. Right, through mitochondria formed into sucrose. That's it. These two are the interlinked. Is it clear? Hello?
Yes, sir.
Any doubt today topic? I think today is last class, right?
Yes, sir.
Yes, sir.
So lipid completed, carbohydrate complete. And what is the fifth unit? Amino acids remain. Proteins. Yeah, eighth unit already completed. Structure of membrane already completed in offline classes. DNA completed. Only 3 units are remain. Right? Hello? First
Amino-
unit
Yes, sir.
in our syllabus to be completed. Unit 1, principles of thermodynamic. In black 2, fifth unit amino acids. And unit 6th protein. Except principles of thermodynamics, amino acids and proteins will be covered in practical training classes. Because in practical, your fourth paper practical about amino acids, about proteins. ఈ అమైనో యాసిడ్ ప్రోటీన్స్ మీ ప్రాక్టికల్ లో ఉన్నాయి కాబట్టి ప్రాక్టికల్ నేర్చుకునేటప్పుడు 2 యూనిట్స్ అయిపోతాయి, 5th and 6th. As I said earlier, principles of thermodynamics, I will give the class with chemistry sir and the time of practical training classes. I will get it done, okay?
Okay, sir.
Any doubt? Today topic, our entire topic, the entire self, biochemistry, physiology. Okay, clear?
Yes, sir.
Can I leave?
Sir, selective questions, sir.
Important questions.
Practical classes రండి అక్కడ చెప్పేది. First you read all. When you need important question before the time of exam, right?
Okay, sir.
First let you read all the questions. The entire back we have to ready for competitive level, but not theory exam. That's why you should focus on the entire book, entire course. The time of exam, I will give you the important questions.
Okay, sir.
Okay,
Okay,
right.
sir. Thank you, sir.
Can I leave now?